then we move to the second part of photosynthesis called the Calvin cycle. Now notice this is called the light independent or the dark reaction. Why? Because solar energy is not a factor here. We don't need solar energy for the Calvin cycle to occur. We can undergo the light reaction during the day, and if necessary, wait and undergo the Calvin cycle at night. Because all we need for the Calvin cycle is the ATP and the NADPH, the chaperoned electron, from the light reaction, and carbon dioxide. Well, carbon dioxide is in the atmosphere whether it's day or night. Notice we're also moving now from inside that thylakoid membrane where we made these products to out in the fluid, the stroma of the chloroplast. The Calvin cycle is referred to as C3 photosynthesis. We'll look at where it gets its name shortly. But there are 3 stages to it. We want to fixate carbon dioxide, then we want to reduce it, remember that's going to be gain electrons, and then we want to regenerate the enzyme that allows it all to happen. There's a cycle in your book. If you'll find your image of the Calvin cycle, it will help understand to see the picture. There's also one in your notes if you prefer to look at it. So we're going to start the Calvin cycle with the carbon dioxide that we brought in from the atmosphere through our stomata, which are the openings in the leaf. Well, when we bring carbon dioxide in from the atmosphere, we're going to attach it to a molecule called RUBP, which is a 5-carbon molecule. So this one has 5 carbons, and we want to add it to carbon dioxide that has 1 carbon. Therefore, we're going to get a 6-carbon molecule. Well, the problem is we just don't have too many 6-carbon molecules, and this one is very unstable. It's so unstable that it rapidly splits itself into 2. It splits itself into 2 3-carbons. They're smaller, they're more stable. These are called 3PG. It's the proper name for the 2 3-carbon molecules. So one more time, we bring carbon dioxide in from the atmosphere, attach it to this 5-carbon RUBP, which always exists in the stroma, the fluid of the chloroplast. These 2 join together, become unstable, therefore they split into 2 3-carbon molecules. Now, what would cause this to happen? What would cause carbon dioxide to join? It's going to be an enzyme, RUBP carboxylase. This is an enzyme because it ends in ASE. We now say carbon dioxide has been fixed. We have taken carbon dioxide and fixed it to RUBP. Step 1 of the Calvin cycle. Step 2. Now this 6-carbon molecule, which split itself really quickly into 2 3-carbon molecules, is going to need reducing, meaning we need it to gain an electron. So here's one of our molecules. Remember, there's actually 2 of them. Here's one of them. This 3-carbon molecule, called 3PG, is going to be reduced. We're going to cause it to gain an electron. It's reduced then to a product called BPG. Now to do that, I got to have ATP. So therefore I'm going to use the ATP that came from the light reaction. BPG then turns into G3P. A lot of numbers and a lot of letters. So stop, look at it a couple of times until the letters at least are straight in your mind. Right, so BPG makes G3P. How? Well, I need an electron to do that. So my chaperone will deliver an electron and then go back to being an enzyme. That enzyme will be recycled by the body to be reused later. So here's the ATP from the light reaction. Here's the chaperoned electron from the light reaction. The ATP does the first change for us, and the chaperoned electron makes the second change for us. So at this point, we have turned carbon dioxide into G3P. How? Let me go back and remind you. We took carbon dioxide, we joined it with RUBP. That was so unstable, that 6-carbon thing it made, that it quickly split itself into 3 carbon molecules called 3PG. And now we're back to here. 3PG becomes BPG, And these do stand for long scientific names. If you'd feel better with a long scientific name, you'll see that it's quite complicated. Glyceraldehyde 3-phosphate. You may be more comfortable with just learning the names. BPG turns then to G3P. Remember, this is happening twice. So in the end, I am getting G3P. But here's the problem. This is a cycle, indicating it should go like this, not like that. And so far we're going like that. We've taken carbon dioxide, 6 carbon to 3 carbon to BPG to G3P. We haven't cycled, but we have fixated carbon dioxide. We have reduced carbon dioxide. So we've done 2 steps. For our third step, we need to regenerate the enzyme that allowed us to start this process. We need to regenerate the RUBP carboxylase. So here's that image. Notice that this is putting all 3 cycles at once. 3 carbon dioxides enter. We fixate it by adding it to RUBP, so I get the 6 carbon. It's very unstable, breaks down very quick into C3. To do that, it's going to take ATP to turn it into BPG. BPG becomes G3P, and I'm going to let go of 1 of them. The other 5 are going to be used to remake my RUBP that I need to start the cycle over again. This is a continual cycle going on out here in the stroma, the fluid membrane portion of the chloroplast. At the end, notice I have made G3P, and G3P will become glucose or other organic molecules, fructose. But it makes that carbohydrate that we wanted to make from carbon dioxide and water. Our 1G3P that was just created is not enough to make glucose by itself. Note it actually takes 2 G3Ps to make a glucose. So if we think back through it, the Calvin cycle has to turn 3 total times to make 1 G3P, and we have to do that twice in order to make a glucose, because G3P plus G3P equals a glucose. So it actually takes 6 total turns of the Calvin cycle to make a single glucose molecule. It's one reason why there's so much ATP required to run our basic processes. It's going to take quite a bit of ATP for every turn. We're going to have to turn 3 times to make a G3P. We're going to have to do that process twice to end up with a glucose. So 6 total turns of the Calvin cycle to get 1 usable glucose molecule. So what's so important about G3P? We can pretty much make it a lot of other things. We can make it a fatty acid in a glycerol. We can make it glucose, fructose, starch like in a potato, cellulose like in a plant wall. We can make amino acids out of it. G3P is the framework to make all of our important carbohydrate products. Now, this is referred to as C3, 3 carbon molecule. That's what we wanted to make. This goes on in the mesophyll. So back to my plant. Remember, I have that waxy cuticle. I have the epidermis. Then I have those layers of plant cells throughout the middle. This was the mesophyll. However, C3 plants undergo a process called photorespiration, meaning in the presence of light, I respire. I bring in oxygen and take out carbon dioxide. That is not what we want a plant to do. We want a plant to bring in carbon dioxide and give off oxygen so we can take it in. Well, the problem is when weather's really, really hot, we close the stomata, the opening, so therefore the oxygen can't get out. So even though we were bringing in carbon dioxide, we were undergoing the Calvin cycle, we were making oxygen in the light reaction, that oxygen's trapped inside and the carbon dioxide's not coming in. So we've shut off bringing in the carbon dioxide and letting out the oxygen. So therefore photosynthesis continues because there's still light. So when we get over here to this Calvin cycle and we expect carbon dioxide to come in and join with RuBP and make 3Pg and make BPG and make, instead of having the carbon dioxide to begin with, all we have is oxygen. So oxygen will combine with the RuBP, and then carbon dioxide will be given off. That is wasteful. That is wasteful in dry, hot weather because we need the reverse to happen. Wheat, rice, oats, Kentucky bluegrass, azaleas, maples, plants that we are familiar with here in the South are our C3 plants. They can, unfortunately, when the weather's really hot and really dry, undergo photorespiration, which is wasteful, and they don't produce as much G3P or carbohydrates for us. So C4 is different because here's the C3 plant that we had. There's our waxy cuticle. There's our stomata to open up and bring in. Here's our plant cells. Here's our vein to bring in water. Well, a C4 uses a different arrangement. Instead of needing water to come in from here and diffuse through all of these cells out to here and potentially lose water out to the environment, this water comes in here and bam, goes straight to the cells where photosynthesis is happening before it ever gets even close to a stoma that might allow that water to escape. In a C4 plant, we actually arrange the cells around the bundle sheath, which is the name of the cells around the vein. Therefore, it can make its C4 product before it gives off the carbon dioxide. In a hot, dry climate, it's going to make 2 or 3 times the amount of photosynthesis as a C3 plant because it's not going to photorespire. It's not going to shut off that stoma and trap the oxygen inside so that the oxygen becomes a part of the Calvin cycle. Instead, it's going to use something called PEP carboxylase, which can't join with oxygen. RuBP can join with oxygen. PEP carboxylase cannot. Much, much better for hot and dry climates because it's a more efficient photosynthesis for those areas. Sugarcane, corn, Bermuda grass, crabgrass all undergo a C4 photosynthesis. It withstands hot weather better. But then we have really hot weather. We have desert-like regions. In a desert region, neither one of those previous 2 types of photosynthesis would be beneficial. Instead, we use something called CAM photosynthesis, or crustacean acid metabolism. So here's what we have different. We have plain old C3, vein, mesophyll cells. We have C4, vein, everybody huddled around the vein. And then we have a C5, which says, you know, the arrangement is not as important to me as the time of day in which I try to do these processes. Because in the daytime, when the sun is out, it is very, very hot. So whereas a C4 plant splits things by space issues, you know, using those cells arranged around the bundle, the CAM plant says, you know, I need to do this differently by time. At night, when it is cooler, I'm going to open my stoma and bring in my carbon dioxide so that I can fixate carbon dioxide at night and not worry about losing anything out to the heat. Carbon dioxide comes in, joins with RuBP, makes a 6 carbon. That splits very quickly into 3Pg, becomes BPG, becomes G3P, 1 goes out, 5 make the loop around to recycle the RuBP, and it continues. So this is going to split it up by time frame. Therefore, it's going to need to bring in the carbon dioxide at night. But remember, for this to continue, for this 3Pg to become BPG, I need ATP. For this to become this, I need NADPH. These came from the light reaction. I need sunlight. So what it's going to have to do is bring carbon dioxide in and store it. it's going to store it inside of a large vacuole. Then during the day, when sunlight comes in and energizes my electrons, I will make my two products, shut down my stomata so I don't lose water, and then at night continue on with the Calvin cycle. Very good for hot, arid, desert-like regions. So each one of these methods has goods and bads. C3 is going to be better adapted where it's colder. A little bit further north from us, more like Kentucky, New York, would see more C3 plants. We do have a few C3 plants here, but they don't withstand our summertime climates. If you plant ryegrass in the winter, which is a C3 plant, it'll be pretty during the winter, but when the summer comes, it's going to absolutely dry up. It just doesn't have the ability to withstand that. So it likes a high moisture, cold region. A C4 plant, better adapted to high light intensity, high temperatures, limited rainfall, us. Cam plants are better adapted where it's really, really arid, where it's very hot, very dry. Even though a cam plant is a desert plant, keep in mind that there are desert plants that are flowering. So we've looked at flowering plants, we've looked at the chlorophyll, how the chlorophyll can absorb the solar energy and energize the electron. We looked at the two processes of light reaction versus the Calvin cycle, and we compared C3, C4, and CAM photosynthesis. It will be beneficial to take the diagrams at the end of your notes and review back through them. When you can label those diagrams without the assistance of your textbook, then you will understand photosynthesis. You may want to organize the diagrams based on what comes first, what comes second, and so on. I would print multiple copies of your diagrams and continually label them and talk yourself through the process until it clicks with you.